Start it again. So our next speaker is William Parker from Duke University. He's going to be talking about the dependency of the immune function on environmental components.
Thanks very much, Mark. I think you can hear me okay at the back of the room. If you can't, okay, we're good. I'll talk to you a little bit today about how parasites interact with the immune system in a way that we think is very important for the health of the host. And so in that sense, there's sort of some question as to the actual definition of a parasite. We'll get into that in some detail. Most of my work has been done looking at wild rats. We won an award for this from Popular Science back in 2009. It's not on my CV, but it's our claim to fame with that work. We've been looking at the immunity of wild rats for about 7 or 8 years now. I think we can learn a lot from the wild rats. So I'm in a medical center, so we're very interested in human disease. We're not so much interested in ecology, but I think I'll be able to hopefully give you something that you'll find useful and helpful for what you are interested in, the ecology of immunology. I'm not an ecologist, not done any work on ecology or parasitology for that matter. I'm a basic immunologist by training as a postdoc. But I think we can use what's going on in post-industrial society as kind of a model for what might happen if we took wild animals and we removed, whole swaths of their, what we would call the biome. And in this context, we're talking about the biome as the actual body of the mammalian host. So we're working on mammals, we're interested in humans, we use rodents as a model for those. I don't know anything about insects, except for I don't like being bit by them. That's about the only thing. So the idea here is that in post-industrial society, because we have toilets, because we have water treatment facilities, we've disrupted the life cycles of some of the organisms that have been with humanity since humanity has existed on the face of the planet. And again, I'm not an ecologist. This is Sarah Perkins is the ecologist, parasitologist that we work with, and I don't torture my friends this way. She sent me this picture just in the middle of a rainstorm. Some of you know Sarah is helping us out with this project and the design and how we analyze various parameters associated with the animal. So in the medical world and post-industrial society, and somebody mentioned it earlier, in the immunology department, we care mostly about the immune system rejecting transplanted organs, and we care a lot about the immune system causing autoimmune diseases and allergic diseases. So I think I'll go ahead and use a more traditional definition than used previously, Andrea. In this case, we're talking about autoimmune diseases when tolerance, in the traditional sense, breaks down and you attack your own body. Your immune system attacks your own body. And by the same token, an allergic reaction is a certain type of immune response where you attack a harmless environmental antigen, say a piece of ragweed pollen or an antigen from a dust mite or from a cockroach. Those are some very, very common. And it's becoming more and more common to attack antigens from food, such as peanuts, or antigens derived from the peanuts. So the idea here is that we're missing some components from our immune system. Graham Rook is a clinician scientist based in London that's done a lot of the work on this. We classify these as in three different categories, or he has, and it's a good classification system. It holds up well. And these are the kinds of things we think it takes to make your immune system stable. And here we're not just thinking about things differently. We're really thinking about different things. We're not concerned about the pathogens or infection. So when we talk about a parasite, we're really only interested in those parasites which we're extremely well tolerant to use the previous speaker's parlance. We're not interested in the kinds of infectious organisms that kill people, that result in substantial morbidity and mortality. So of the of the more than 100 different species that are specific for humans that are classified as parasites, we're probably only interested in 3 or 4 in terms of utilizing them to help us get our balance back. And I'll talk about that in a bit. But I think the first thing I'll do is we'll look at the microbiome, or the mutualistic bacteria that live in our body, and sort of take as an example that connection and how that works. Let me get my time set here. Try to end in 35 minutes. So there's long been an issue that we really don't know how our immune system treats those beneficial bacteria that are in our own bodies. And the world famous immunologist, the discoverer of TLR4 sitting back there, showed you a slide that says, you know, that we block them out. That's what we do. We produce IgA, we produce mucus, and that's been the traditional paradigm for a long time, is that we're kind of running a constant blocking action against the bacteria in our own gut. And those ecologists in here, you already know, you don't even have to think about it, it doesn't make a lot of sense. But that's the classic paradigm in immunology. That's what's held up. So and the classic paradigm is really came about from a paper in Science in the early 1970s. The idea being that immunoglobulin A and mucin, and here in the little diagram we've got the IgA, or the little bow tie molecules, basically agglutinate or stick, cause the bacteria to clump together. And the mucin molecules sort of run a blocking action here along the, this is an epithelial cell, or a cell that lines the gut, and the little fingery things are the microvilli. So this is a single cell. The cell would be very large, maybe the size of this room or something, but this would be the surface of the cell, microvilli. The cell's producing mucus that keeps the bacteria away, and then the mucus and the IgA together clump the cell up. This was based on some experiments that were done in a test tube, where you take some IgA, you incubate it with some bacteria, and it clumps the bacteria together, and what you find is that they don't stick to cells very well after they're clumped together. Right? It turns out it's an attractive idea. It made sense to a lot of immunologists for a couple of reasons. One reason is because when you see a patient who's in the hospital who's very old and sort of has they're just dying because of senescence, and they're having multiple organ failure, usually what gets them is the actual bacteria in their own gut, because as things break down, the heart, as kidneys start to fail, this bacteria are released and they result in death. And we also know that the secretory IgA are made against pathogens as well as beneficial bacteria, which again led most people to believe that what was going on was that the secretory IgA and the mucus were antagonistic to our own beneficial bacteria. This is one of the first experiments we did, published in 2003. What we did essentially was we were looking at, on this axis, growth of a biofilm, or literally a living film of bacteria, on various different surfaces. In this case, it was on a plastic surface. And we added secretory IgA. We changed the conditions, though. We let it incubate over a long period of time, and we kept constantly washing the system through. So when you had secretory IgA, you could grow a good biofilm. And if you used an enzyme and you cleaved the secretory IgA in half so that it could no longer stick the bacteria together, the biofilm fell apart, and other proteins didn't work to form the biofilm. Now, this is not the only experiment we've done. We've published 8 papers on this over the past few years with hundreds of different experiments, and worked out the molecular details of how the IgA interacts with bacteria. But the point is, if you set the experiment up in more vivo-like conditions, you can see that the IgA can be supportive of bacterial growth. And this is sort of a breakthrough technically that we made. People have been trying for decades to grow a biofilm. This is without magnification, so it's just a little cassette that has human gut epithelial cells growing on it. And over here, we just took a picture of that cassette. And it's black because the gut epithelial cells are essentially transparent. It's a single layer of cells. But when you add secretory IgA to the mix, and bacteria, we put bacteria on both sides, but we only added the host molecules over here, that's when you get a nice biofilm formation. So again, people have been trying for decades to co-culture gut epithelial cells with bacteria in this biofilm format. And the secret to it was very simple. You simply add secretory IgA. You can grow these things overnight. And the mucin works the same way. This is a different format, but again with no magnification. We're just looking at some tubes here that have gut epithelial cells in them. And you can see that with secretory IgA or with mucin or with a combination, you can grow a nice film. And in this, we still have the epithelial cells. We still have bacteria. We just didn't add any host molecules. You can see a little bit of refraction of light off the bottom of that test tube, but there's no biofilm of bacteria growing on the cells. So the model that we sort of developed is one where pieces of biofilm get shed off, and we just this year published a paper, sort of a how shit happens paper. If you look at human feces or feces from any other animal, what you'll find is you'll find little chunks of these biofilms in there, but most of the bacteria are not in a biofilm. It's an equilibrium between planktonic or free-floating bacteria and little pieces of biofilm that get periodically shed off. So that's what's going on apparently. When you go look in the actual animal, this is in a rat, this is in a laboratory rat, which may be different than a wild rat. We'll get to that. But you do find the predicted biofilms, and we had to develop the techniques to look at these biofilms. Classic techniques used in pathology ended up destroying the biofilms, and you see no bacteria at all. And the little dotted things, this is a classic biofilm. It's arranged in a certain pattern that goes with the flow of the solution that's in the lumen of the gut, or in the inside of the gut. And these big things down here, those are this is stained with acridine orange. Doesn't really matter. The point is, these are the cells of the rat host, and then this is the biofilm here. Turns out the biofilms are most concentrated in a rat. They're most concentrated at the apex of the cecum. In human, they're most concentrated in the appendix. A friend of mine who takes out appendices on a regular basis, just based on what I just showed you, thought, I bet I know what the appendix does. So out in what was that, 2007, we sort of made a deductive proof. It's a lot of circumstantial evidence supporting it, but it's a simple deductive proof. We confirmed that the biofilms are the most concentrated in the appendix. We already knew that biofilms are supportive of bacterial growth, so therefore the appendix, where it's known to produce a lot of IgAs, probably supportive of bacterial growth. The overall model that we had something like this, so got an appendix here, kind of a safe house in case this evil-looking red thing gets in here, causes an infection, and you have a diarrheal response, recovery, and then back to normal. The only theory really other than this safe house for bacteria had to do was developed by Charles Darwin as far as what the appendix was for. And this is my last slide on the appendix. This is my last slide on the microbiome in general. The point is that I want to make is that if we think about how the immune system is really interacting with our environment, it gives a much better feel for the biology of things. So the immune system, it's not a defense system. Okay? It's an interface. It's an interface with the environment. So certainly defense is a part of that, and being humans, we're interested in things that kill us. And that makes sense, but most of the molecules that our immune system produces are there as an interface. And I'd be curious one day if we figure out what